Hello, this is lecture number 21 in the series from Albert Lehninger's Principles of Biochemistry, 7th edition. Today we will be discussing nitrogen cycle and discuss the bioenergetics of phosphorylated moieties. The nitrogen cycle is a condition where we reference the transfers between nitrogen, ammonia, nitrite, nitrates, and amino acid done in bacteria and plants. So animals convert the amino acids to ammonia, and the ammonia re-enters the cycle. There's intermediary metabolism, which combines steps in the pathway. Some of these pathways have simple reversals of reactions that occur. Others loop out of the established pathway and achieve their results through a different process. So generally speaking, catabolic pathways are convergent. One product is formed, whereas anabolic pathways are divergent, where many products are formed. Pathways are regulated to attain a certain number of simple different pathways and enzymes are utilized. So when we look at biological regulation, that is dependent on several things in the chemical milieu, namely substrate availability, allosteric regulation by an intermediate, growth factors, hormones, and regulatory factors. By point of review, we again talk about the Gibbs free energy, which is the change of which is equivalent to the change in enthalpy, which is the delta H minus the temperature and then the change of entropy. So spontaneous reactions will have a negative delta G. Heat-producing reactions will have increase of delta H. Cells function at constant temperature. By definition, they have temperature optima, and the reaction of A and B to C and D has an equilibrium constant, which is a product of the multiplier of the products by their moles divided by the multiplication of the reactants by their raised to the power of their moles at equilibrium. We usually factor the water, protons, and ions into the equilibrium constant, and those are not brought into the determination of the right side of the equation. So anything that is associative is grouped into the equilibrium constant. So when you run the ratios, the delta G is equal to minus the gas constant R times Kelvin temperature times natural log of the equilibrium constant. The energy change is the difference between the reactants and products. Standard free energy is delta G naught at equilibrium. And again, if you put in the equation for the equilibrium constant, you can expand the delta G and put in a reference baseline at equilibrium as delta G naught. The free energy changes are additive between the reaction steps. So for the bioorganic reaction mechanisms, it's critical to understand the sequences of the metabolic pathway. Again, you've got atoms and orbitals located in shells S, D, and F. And the acids, by definition, accept electron pairs. Bases donate electron pairs. This is the basis of Lewis-Aspect theory and is fundamental to understanding the various pathways that occur in biochemistry. So the electronegativity increases progressively. So by definition, the highest electronegativity among the commonly encountered atoms is possessed by oxygen, followed by nitrogen, followed by sulfur, followed by carbon, followed by phosphorus, followed by the proton. And this governs the free energy of a given reaction. So a nucleophile is the base. It donates electrons. The electrophile is the acid. it accepts electrons. And these carbon-carbon bond breaks are chemically unfavorable, so you've got to delocalize the electron pair to a nitrogen or an oxygen, even if you have to introduce a nitrogen or an oxygen. The vitamins that I've referenced earlier work as cofactors for these. Oftentimes they will bind and act as the electron sink for the vitamin's nitrogen or oxygen atom. And some of these formulations, such as thiamine, are quite elegant, and we'll be getting into that in due time. The mechanisms are utilized in pathway lectures, and they are internal rearrangements, isomerizations, eliminations, and these electron pairs are moved about to facilitate the reaction. So it's important when looking at these, again, not to look at it as a series of English words or chemical words, but try to look at the structure and be able to think through, how do I get from point A to point Z, and that would make the process much more easy to understand. And there are also free radical and group transfer reactions, including acyl, glycosyl, and phosphoryl groups. Redox reactions occur, and those have an affected change in the oxidation state. Biochemical reactions differ from chemical reactions because the former, the biochemical reaction, does not require redox equation balancing. So that is something that you see in organic chemistry, where you've got to balance the redox reaction with water, with protons, with hydroxyls. Not really needed here. The direction of reaction and value of the equilibrium constant is what's of concern in biochemistry. And the most critical energy entity is adenosine triphosphate. This is a double anhydride ester and is the most critical energy currency model. And this is what we have right here. So you get hydrolysis of the terminal phosphate here, and then this is a high energy link, not because it's represented by a squiggly line, but because this is a double anhydride. 1, 2. So there's an energy yield 7.3 kilograms here, a smaller yield going from ATP to AMP, which is shown here. So to give an example, you have resonance stabilization here, where the oxygen, the oxygen ester is negatively charged relative to the phosphorus atom. You have proton over here, and this is the product of that, which is an ATP, and you can do an ionization of this and get a charge balance. By the time you get to the delta G of this transformation is about 30.5 kilojoules per mole when you get this reaction going, including its ionization. So under standard conditions, this is a 30.5 kilojoule per mole. The ATP equilibrium is usually in excess of that required for equilibrium, and you also have other phosphoryl esters which have also energetic sinks or residues, such as phosphoenolpyruvate and phosphocreatine. And thioesters can be used as an energy source. Acetyl-CoA is the predominant example of that, and that'll be a part and parcel of the Krebs citric acid cycle. So going back to this formulation, here's acetyl-CoA. You've got this methyl group attached to a carbonyl, and the energy component is in this bond here. You can hydrolyze it, get acetic acid, and ionize it to get a splitting of the charge between here with resonance stabilization of the acetate moiety, and then this delta G is 31.4 kilojoules per mole. The ATP, it also performs group transfer, not through hydrolysis. The sum of energies of the individual reactions proceeds as you transfer phosphoryl groups. ATP is also involved in muscle contraction. You can also get another source of energy through electron flow in redox reactions. We'll be covering that. Redox reactions are also used in metal ion transfer and also in dehydrogenation reactions. You can get transfer of electrons, hydrogen atoms, and hydride atoms. This will be seen in oxidative phosphorylation, and that will be another series of talks. So when we look at the various molecules and their energetic components, we can look at alkanes such as ethane right here, ethene, which is an alkene linkage there, ethanol. These are progressive orbitals here, so we want to look very closely at what the electron pairs that occur, and that you have the capacity for carbon to have 4 different moieties attached to it. So studying these, acetylene with an alkyne linkage. This is a triple bond right here. Formaldehyde, acetaldehyde. So studying these very simple molecules gives you an idea of where the electrons are, and the electronegativity, again, is possessed primarily by the oxygens here. So using the oxygen as a driver, say for a carbon that's over here, and knocking the bond, creating here, and then having a negative charge on this oxygen here, and then you can drive an acquisition of a residue by using this, usually coupling with a reaction. So I would probably urge that you look at these electron pairs as carbon monoxide, acetic acid, carbon dioxide right here, and to see what these carbons and oxygens are doing here. And you can use this as a model for ascertaining these pathways later on, and also determining where your energy potentials are. So electron affinity and reduction potential, there's a Nernst equation, so there is an electrical equation that can be employed to it. This has the electric potential equivalent to baseline plus RT over NF, where N is the number of electrons transferred, F is Faraday constant. There's specialized electron carriers such as nicotinamide, adenine dinucleotide, NAD. FAD is flavin, adenine dinucleotide. Generally, you can get 3 ATPs generated off of this process, 2 off of this, and the equilibration between NAD and the reduced form NADH. You find this in about 200 enzymes. We'll be running into quite a few that utilize this energy transfer for reducing and oxidizing compounds. We call these oxidoreductases. And FAD is derived from the vitamin riboflavin, which is a vitamin B2. So when you look at NAD, this is the structure of it right here. In NADP, this number 2 carbon has a phosphate ester right here. There's adenine. So this is NAD right here. And then you reduce it, and you get NADH. So the protons occur at this point, or the stereoisomer flipped to the back. Either way. So this is the reduction that occurs at this carbon here, and the electron pair sinks into this this nitrogen right there. And this is the R group, which is there. And this can be quantified using spectroscopy with a decrease of the absorption at 260 as you go from the oxidized to the reduced form. FAD is roughly the same thing. You can get reduction. This is FAD, this is FADH, which is a semiquinone where you get partial reduction, one proton attaching to this point. This is a positively charged, and you can get a second proton going to the same place, eliminating the positive charge right here. Again, this is the moiety here. FAD is the entire one. There's the adenine, ribose, the flavine component right here, and the upper part of this, with this being the R group. This is where the reduction occurs at this point. First one and then two protons, and this will generate two ATPs. And that will be it for this talk. Thank you very much.